Our next speaker is Francesco Iorio. Please.
Thank you very much. It's not easy at all to deliver a talk after so brilliant keynote, but I'll try not to bother you too much. So, okay, I'm going to present you a bit, at least I'll try to give you a flavor in 10 minutes about a project I've been involved in the last few years while working at Sanger, where I lead a small computational team that calls the Dependency Map Analytics. And this project is funded by Open Targets, which is a private-public partnership between drug companies and academic entities with the aim of identifying new therapeutic targets. So the aim of this project is to really try to accelerate the drug discovery process by providing some guidance in a data-driven way on what are the therapeutic targets that the development of future anti-cancer drug should focus on in the future. So what we've done is basically to use a large-scale CRISPR screen performed on a collection of more than 300 different cell lines. And with this technology, we were able basically to switch off all the genes in turn, one by one, and measuring the effect on the survival of cancer cells from 30 different cancer types. And then we combined the output of this screen with a quite comprehensive bioinformatic pipelines, whose first aim was basically, first of all, to discriminate between core fitness genes from context-specific essential genes. So we don't want to account too much of genes that would kill any type of cell upon inactivation via CRISPR. And then we took the genes that show a context-specific essentiality, and we integrated their fitness score, so which is reflective of their effect on viability upon inactivation, with other functional evidences, like a gene being highly expressed on a given model, or being part of a pathway that is generally essential in a given cell line. And then basically we fed all these evidences into these bioinformatic pipelines, which spits out basically all the gene of the genome ranked from the most promising one for further drug development to the least promising one. And then we fed all this data on a public available platform that calls Project Score. And this is part now of an international effort that calls the Cancer Dependency Map, which is an international endeavor involving the Broad Institute and the Sanger Institute, which aims at put together the first catalog of all the genetic dependencies that might be exploited therapeutically in cancer. So just to give you an idea of the experimental pipeline, so the cell lines were engineered to constitutively and stably express the Cas9 enzyme, and then we transfected a library of more than 100,000 single-guide RNAs. On average, we transfected 5 single-guide RNA per gene, and following selection, the cell lines were grown basically for 14 days, after which we harvested the DNA, we amplified the target sequences of the single-guide RNAs, and we derived single-guide RNA counts. Now, the idea is that if a single-guide RNA integrated into a cell and once expressed targeted a gene that was essential to that cell's survival, then that cell wasn't able to pass that guide to the daughter cell. So you should see a lower representation of that single-guide RNA in the final population. So just by doing a differential representation analysis of the single-guide RNA, you are able to quantify at what extent knocking out a gene reduces the viability of a given cell line. And this can be done with published models like the median variance modeling implemented by Magic, which is not different from EdgeR that is used to do differential expression analysis on RNA-seq data. So this is to give you an idea of the cell line composition. So we included cancer types that are the most prevalent one, as well as cancer types that have a variable response to therapy. And on the right side of this figure here, in this plot, each column represents a cell lines, and you have 3 groups of genes. So the blue group is made of ribosomal protein genes, which are known to be really, really essential and should kill any type of cell upon inactivation. And then there is a green group of genes, which is basically made of another prior known list of genes, which is from shRNA studies. And then the gray group, which is basically genes that are known to be non-essential, like olfactory receptor genes or other genes. And as you can see, there is a very nice separation between these 3 groups, and even when the phenotype on the extreme right is not that strong, they appear to be quite well separated, meaning that you can fit to distribution, and therefore you can predict the role of other genes that do not belong to this set. So this is a landscape of the results. So we had a median number of 1,500 essential genes per cell lines. And as you can see on the right, in that plot that shows the number of fitness genes exerting their reduction of viability in a fixed number of cell lines, the vast majority of the genes is essentially in few cell lines. So they appear to be context-specific. So what we did, we put together this adaptive statistical method that calls the adaptive DAISY model, which was able to identify a more robust set of human core fitness essential genes, which with respect to other state-of-the-art set of essential genes that were published so far, seems to be more robust and covering better gene classes that are known to be essential, involved in fundamental processes, cellular processes. So, but most importantly, this method is able to discriminate for each cancer type group of genes that are core fitness and group of genes that are context-specific essential. And here you can see on the left plot here the number of the genes in each of these two classes across cancer types. And consistently with their role of core fitness genes, they have a stronger effect on viability, right? So if a gene is predicted to be a core fitness gene, generally the effect on viability, on reduction of viability, is stronger than the context-specific one. But most importantly, there are these very interesting cases of genes that are context-specific essentials. So they kill really, really strong, a very reduced number of cell lines. Okay, so those are the most interesting target now. And the next question that we asked was how we can define the context, the genomic context in which this gene seems to be so essential. And what we did, we used a manually curated collection of about 700 clinically relevant features encompassing mutated genes, mutated driver genes, copy number amplified segments. And then we did a systematic correlation analysis of the status of these features in the cell lines with the essentiality of the context-specific genes. And of course, we detected established and expected cases like an increased essentiality of NRAS when NRAS is mutated, or increased essentiality of RBB2 when RBB2 is mutated, but also a number of novel and interesting cases. So what we did, we took all the results from the systematic and statistical test, and we pulled it together with the fitness effect of the genes and other functional evidence that I mentioned before, and the result is basically shown in this figure here, where on the left you have those targets that we deemed as high priority. We define a proper statistical threshold for priority. So this is the 624 high priority target that should be definitely looked at for the development of new anti-cancer drugs. And on top of this, we overlaid information about the target tractability, and we split the targets into three groups. The first one is made of genes for which there is already a drug, at least in preclinical development, which basically offers opportunities for drug repurposing. And then a very interesting group here is in the middle, group 2, and it's made of targets that are not currently druggable, but there are evidences of future tractability. And then there is a third group of targets that are not currently druggable and for which there are there are weak evidences for future tractability. And as you can see there is an enrichment for transcription factors and DNA binding proteins. So what we did, basically, we picked two of the best cases in ovarian and colorectal cancer, this Werner gene that ranks really, really high comparably to KRAS in colorectal cancer, and in ovarian as well. And basically, we put it forward for further experimental validation, finding that this gene, the marker for there is a strong marker for this gene, for the essentiality of this gene, which is the macrosatellite instability. So this gene is particularly and specifically essential in macrosatellite unstable cell lines from ovarian and colorectal cancers. So we proposed this as a new target for this subtype of cancers. And we further experimentally validated here with a clonogenic assay to prove that the effect is specific to MSI in stable cancer. And then we put it forward for in vivo validation. So we implemented a single guide RNA inducible expression on a cell line, and then we transplanted these cell lines in mice. And you can see here that upon the activation of the expression of the single guide RNA, you have a shrinkage of the mice tumors only in the mice that are treated with doxycycline, which is the agent used to induce the expression of the single guide RNA. Okay, final slide. All these results are publicly available and queryable on the dependency map, DepMap score website. And for the future, we are integrating this dataset with similar dataset from the Broad Institute, the Project Achilles, and consolidating other omics. We aim at putting together the first map of all the cancer dependencies in 1,000 in vitro models. And this is led by a postdoc in my team who calls Claire Pacini. Good. I need to thank a number of people that were involved in this project, particularly Fiona Behan, who performed all the experiments, and Gabriele Picco, who did the in vivo validation, and Open Targets, who is our funding entity. And there will be a live demo session during which you can put your hands on this data, and this is at the EMBL EBI stand on Wednesday afternoon at 16 p.m. Thanks. Thank you.
I think one of the challenges of cancer screening is resistance. So when you do the screening, so can you comment on like how prioritizing is actually associated with potential
Emergence of resistance. Yeah. First of all, yeah, one thing that you could do is to account of these. If there are clinical trials already established for at least one cancer type, you could account for this into the prioritization scheme. Or you could perform drug plus CRISPR screens and see, in a resistant model, for example, and see which gene, when knocked out, they would rescue the sensitivity to the cell. But at the moment, with this method, we only account for intrinsic drug response, so not acquired drug resistance. Good. Sure.
Maybe we can discuss. You can discuss during the breaks. Thanks.